Regulation of contraction by calcium ion. Calcium ion is the intermediary signal that allows striated muscle to respond to motor nerve impulses. The calcium ion signal is correctly interpreted by muscle only when tropomyosin and the troponins are present. Specifically, actomyosin, prepared from pure preparations of actin and myosin, thus containing no tropomyosin and troponins, contracts when ATP is added, even in the absence of calcium ion. However, actomyosin prepared directly from whole muscle contracts in the presence of ATP only when calcium ion is added. Clearly, muscle extracts contain a factor that confers normal calcium ion sensitivity to actomyosin. The factor is the tropomyosin-troponin complex. Actin-thin filaments consist of actin, tropomyosin, and the troponins in a 7:1:1 ratio. Each tropomyosin molecule spans 7 actin molecules lying along the thin filament groove between pairs of actin monomers. In the absence of calcium ion, troponin 1 is thought to interact directly with actin to prevent the interaction of actin with myosin S1 heads. Troponin 1 and troponin T interact with tropomyosin to keep tropomyosin away from the groove between adjacent actin monomers. However, the binding of calcium ions to troponin C appears to increase the binding of troponin C to troponin 1, simultaneously decreasing the interaction of troponin 1 with actin. As a result, the tropomyosin slides deeper into the actin-thin filament groove, exposing myosin-binding sites on actin and initiating the muscle contraction cycle. Because the troponin complexes can interact only with every 7th actin in the thin filament, the conformational changes that expose myosin-binding sites on actin may well be cooperative. Binding of an S1 head to an actin may displace tropomyosin and the troponin complex from myosin-binding sites on adjacent actin subunits. And that's the regulation of contraction by calcium. Have a great day.